Okay, so now we have pre-processed BAMs with aligned read and the base quality scores are calibrated and the duplicates marked, and we're ready to call some variants. So here we'll just talk about the overall different pipelines we have for calling different types of variants and the actual output formats that the variant callers will give us. So just a general kind of idea that so the human genome has roughly 3 billion sites, and any two humans will share most of their DNA together. And most of the variation in the human genome and in each particular one of your samples will actually be at the common sites, and it will be shared between individuals, and only like a small proportion will be from de novo variants or rare variation. So how do we actually represent the variant calls for one particular sample? So we don't really want to just show what the genome will look like as a fully assembled thing. We want to just tell you what is the difference with respect to the reference, right? What are the difference between if there was a variant at a particular loci, what is that variant, and what was the actual base in your human reference. So reference is one of the most common resources in our analysis, and it's important to keep track what that reference is. Sometimes when things go badly or tools fail unexpectedly, a very common thing that happens is that you just have a different reference resource. And even for the same build of reference, there are different kind of different reference files that are slightly differently formatted. The reference that is the most common now is HG19 that came out in 2009, and the the most recent reference is GH38, which came out recently, and that has some improvements towards the GH19, and it's actually has sequences in the places where GH19 did not have, with near the centromeres, for example, low complexity regions, and it shows the different. It actually shows the different haplotypes of the population, not just one most common haplotype that occurs. So yeah, the data that aligns to GH38 is of somewhat better quality than GH19. So there's a few types of variants to consider when we're doing analysis. A lot of our talks will be centered around short variants such as SNP and indels. And for indels, the size that we consider them to be indels is around less than 50 base pairs. And basically for anything more than 50 base pairs, the haplotype caller is not responsible for. Then another group of variants is copy number variations that we will also talk about, where the part of your DNA is duplicated or deleted. For example, on the left you have just no reads at all, where you will have a, what's called a homozygous deletion, or, well, you can also have one copy of your, one strand of your DNA deleted, one copy of your DNA deleted, in which case you have a homozygous deletion. And, well, you can also qualify the copy number variants as part of overall structural variation kind of family of variants. And the other variation that structural variant include are things like inversions, translocations, and also a lot of different subtypes and combinations that we will talk about later. And finally, as a side note, we also have tools for determining pathogens in your DNA, and we have a tool called PathSeq that takes care of that for you in case you are in a situation where you think that you might have some kind of contamination in your sample. And finally, for each type of these variants, we have different pipelines for doing germline analysis or somatic analysis. Okay, so this is what the variants actually look like when you look at the BAM file in IGV. On the left right here, you have very likely insertion, a little indel right here, where you can see that there's a half of the read supporting it, so it's a heterozygous site. There's this G to C SNP right here. Then there is another SNP. This is a het SNP right here, and finally you see there's this huge mass of misaligned reads, which possibly points to some kind of structural variation. And what kind of structural variation is actually pretty hard to see, but that's what it most likely indicates. Right. There's no example of copy number variation, but the general kind of idea is that if you have a copy number variation, you expect some change to the coverage profile, and there's your coverage profile for the sample, right? Coverage distribution over these bases, and you expect if you have some, for example, deletion, if it's a homozygous deletion, your coverage will just drop to zero, and if you have homozygous, it will be roughly half. It's much more difficult to see from exome samples because the coverage profile is really non-uniform. Okay. How many people are familiar with VCF format? So yeah, basically most people. And so briefly, you have every VCF has a header and records, and header has information to describe what fields you have in your records, and each line of the VCF past the header indicates one single variant with position, the reference allele, the alternate allele, quality, whether it was filtered or not. And then there's the info column that overall has metrics on that variant, and then there's genotype fields for each particular sample you have, and the format of the genotype field is described in the format column. So if you want to, for example, know what this guy in the genotype field represents, you go to the format field that says DP, and you go to the header and you see, okay, it represents read depth. What else? So in the very first attribute of the format field is the genotype, and you can actually see what the genotype is here, right? So here the reference is G, the alternate is A, so this is 0, 0, meaning it's just a reference. And this is 1, 0, so it's a heterozygous site. And we can also represent phasing information by if the variant was phased, so like we know which variants came from which contig, which then this will be not the dash line, but it will be a straight dash line. We can also use, so this was for representing variant information of SNPs and indels, but we can also use VCF to represent structural variations, even though it gets a little bit more messy. So the idea is the same. You also have your reference allele, which in this case doesn't really tell you anything, but then you have alternate allele that gives you the actual variation that is in your field, and then the info field encodes what type of the variation you have, and also so because you only have the position, the starting position of your variant, you also have to put the ending position because structural variant has large size. So you put it in the info field. So you have the start right here and right here. And finally, there's also a way to indicate kind of arrow bars on where your structural variation begins and ends. And this is encoded in these two info fields right here, CI end and CI pose right here. So, so you can also use VCF to output variants of the structural variation pipelines, which we do. So this is the pipelines for which we have best practices in GATK. A lot of the focus will be on the haplotype caller. We will also talk about the somatic version of it, which is GATK4 that is currently have been over very heavy development. Then we have a set of copy number variation tools, both for germline and somatic. And we will give a talk about the somatic pipeline with a lot of details. There's not going to be a dedicated talk to germline copy number variation, but this is actually the tool that I am responsible for developing, maintaining. So if you guys have any questions, feel free to come and talk to me. And we also have some tools for doing more general structural variation calling, which we'll also shortly touch on. So first, SNP and indels. So one of the things about just doing variant calling in well, SNP and indels world, but also in other types of variants, is that we want to call sample jointly. That gives us more power, more information on, well, first of all, it gives us the population frequency, which will which help researchers in doing their association studies and so on. But it is also helpful in actually giving you more power, more information to call variants with more confidence. Because, for example, if you have just one single sample and one single site with low coverage, well, you're not sure really whether this site is prone to some particular artifacts of DNA sequencing. But if you have 100 samples, then you have more data at that particular site, so you can say, well, probably it's a true variant, or maybe it has a lot of artifactual reads and so on. And then you can also apply information from your family family sequences to determine de novo mutations, and also give you better confidence over your calls, and the ethnic background to also improve your call set. Okay, the way it is done in haplotype caller is by having this intermediate output called GVCF, or genomic VCF, where instead of just encoding all the positions where you have variations, you also have all the intermediate positions where you don't actually have a variant, but you still want to have a representation for your confidence of the fact that you don't have a variant there, right? You want to somehow show that, well, there could have been a variant there, but probably not. And so you combine different samples in this way. So you basically, in the GVCF, you have a field for roughly every position of your genome, and that gets and then you do joint calling using that large GVCF. Another advantage of having multiple samples is that you can do variant filtration. So if you have a lot of data, you can see what kind of attributes of your variants will correlate with bad calls, and you can have machine learning models that will filter out bad variants. That they will determine which variants are bad and which variants are good. This is an example of a plot of two different attributes. It's not really important which ones, but the idea is that this bulk right here of variants is good, and then you progressively get worse and worse variants as you get farther away from it. And we will actually discuss the idea behind variant filtration in talks tomorrow, and our very new solution to it that includes deep learning. Yes? The axes are two different annotations from the VCF. I don't know which ones, but we use, in the VQSR, for example, we use 7 of them, right? These are just two ones that give you pretty looking plot. Yeah. It's just an example to kind of show the idea behind variant filtration. Finally, using population information, we can refine our genotypes. So for example, like if you have a sample we refined 417, there's a sample that where we found 417 de novo variants. We really don't expect there to be so much, but we can take advantage of population priors and optionally family priors to kind of refine that. And while you have a variant and maybe you're not really sure in it, and also you know that it's not very commonly occurring in the population, then you can adjust your confidence based on that fact, and you can filter it out. So we can filter out these 417 variants to only 8 high confidence variants, which is much better. Another tool that we have in GATK is Funcatator, so it's a functional annotator revariance, and it will basically classify your variants into what is their functional purpose, whether they're an intronic region and do not really have as much, they don't affect as much your transcription, or maybe they're just synonymous coding change that does not change your protein, or maybe they're non-synonymous, and so on. And so Funcatator annotates your VCF and produces the classification of each variant. Right, so this is the Germline and Indels workflow. So Bhanu talked about this pre-processing stuff. Oh, no, never mind. That pre-processing stuff comes before that. So this is the haplotype caller, finally join calling, and this is the variant filtration step that I just talked about. Okay, so that was the germline SNP and indel calling. Next set of workflows that we will talk about is the somatic variant of that. And the problem that we're trying to that we run into with somatic samples is that we have very different signal to noise ratio, because besides the sequencing problems that we have in our germline samples, well, in addition to that, we have the contamination with normal, so we don't actually have the when we extract the sample from the DNA, we don't just grab the stuff that's heterogeneous tumor. We probably grab some stuff with normal sample, some stuff with the tumor that we're interested in, but also these pre-tumor cells that will actually have different, slightly different variants than our bulk of our tumor. So that's the heterogeneity factor right here. And so the signal to noise ratio will be much smaller, and we also do not have the same assumptions about what variants look like in our tumor samples. We don't have the, we can just assume that half of the reads will have a variant, right? We're looking for things with very low allele fractions, which makes the problem much harder to model. And the way to get around it is to, well, again, we use more samples, right? We can use the matched normal sample, right, for figuring out which variants came from germline, because we don't want to include that in our somatic analysis. And we can also use a bunch of samples to subtract the systematic noise from our tumor sample. Those are usually normal samples, and we take a bunch of them, and that's what we call panel of normals. Panel of normals is a resource that helps us get rid of the noise. And it's the same name for a lot of different things in a somatic SNF and indel calling. It means one thing, and in copy number variation, it's slightly different thing. It has the same name though. Yeah, so as I said in the previous slide, we usually have the luxury of having unmatched normal sample when we do tumor, when we do somatic analysis. So we can see that, well, this variant right here is also in germline, so, well, we're not really interested in that. However, this variant right here, it's in somatic sample, in tumor sample, but not a germline, so it's probably a somatic variant. And the panel of normals, the one that we used to get rid of the noise, is used for subtracting out the artifacts of the sequencing itself. Okay, so this is the big MuTech2 pipeline, where we'll discuss it in more details on day 3. It has other details that I haven't mentioned for doing more intense filtering and also figuring out cross-sample contamination. So we will discuss that tomorrow. Mm-hmm. Yeah.
So when do we go to join calling of somatic samples?
Like when I was asking when it will be developed, or
yeah.
I have heard some chatter that there is a project to do that. Oh, did I not? I've heard some chatter that there's a project to figure that out, like how to do joint calling and somatic samples, but that is not something we currently do.
Why you haven't picked us already?
Well, I mean, it starts kind of at the conceptual level of why do we do joint calling, and what's the underlying kind of model, and what do you get out of joint calling? Because joint calling imposes some computational costs that you don't have if you're just processing samples individually or just in tumor normal pairs. What you get out of it for germline calling, and we'll go into a bit more detail in the next talk, is that you're taking advantage of the fact that most germline variants are present in more than one person. And so when you're calling variants that are present in more than one sample, you get some confidence boost, some additional evidence that allows you to make the call with more robustness. And so that's one of the key things that we take advantage of in germline joint calling. But for somatic calling, that's not necessarily the case, because you have a lot more. I mean, in some cases, you might have the same variants, but in many cases, you have different variants that just cluster in similar genes, things like that. You have something to say? Go ahead.
What's the recommend? Does GATK has a best practice? Somebody's interested in investigating the clone evolution.
Yes. Let's.
We are actually working in on that problem, but using the copy number variation approach. So we're trying to develop some tools with that.
At least in the algorithms that we are using to estimate the clone evolution, we must have the variant frequency, so it shows the somatic variance in each sample.
Let's.
There we have to do genotyping.
Are you going to be there tomorrow? Actually, Thursday. Let's talk about that more in detail Thursday, because we're going to go over the algorithms involved in a lot more detail. And I think that will be a more appropriate context to discuss this, since it's a little bit more of a specialized question. If that's all right? Cool. Carry
on. Okay, next up are somatic CNVs. The idea here is that we're looking at the coverage changes and a coverage profile to determine whether the variation occurred. So if we have more reads that got mapped to a specific location, that is a signal for copy number variant. Let's see. Yeah, that's basically what I want to say. So you have the copy number variant pipeline for doing somatic analysis. That the main kind of step here is to denoise the coverage, to normalize the coverage, because especially for exome samples, if you just plot this coverage profile for exome samples, you really will not have any signal. So the biggest concern is normalizing that. And finally, you have the segmentation step where you determine the boundaries of your events. So here, and the problem for copy number variation is a lot different in germline and somatic. It's actually, in a way, more harder in germline because the size of the events are very small. So you don't have this huge signal where your entire chromosome gets duplicated. You just have like very tiny, maybe a few megabases long events, but not an entire chromosome. Yeah, so there's several steps. There's several steps of the denoising, followed by segmentation, and we also take advantage of the allele, the SNP information in order to do segmentation. Yeah, this is overall pipeline. We will also talk about it more detail on Thursday. Finally, we have more newer method in development in the SV world and Germline CNV. And for Germline CNV, we have a more or less kind of stable stable pipeline. It's still under some development. Some command line options may change. However, it's more or less in a stable condition. So the as I mentioned, calling in germline samples it's a different flavor because the size of the variance is much smaller, and so the problem. So before in a somatic pipeline, the kind of denoising and the calling are two different steps because we don't need to have so much sensitivity because the variants are so large. However, here our model kind of needs to know about those two things simultaneously to be able to pick out these small events. So yeah, so we have a really fancy model for doing germline analysis. Welcome to try it out. We have a tutorial about it online as well. And so finally, structural variant calling. So there's a lot of different flavors that come in. Here's some examples. So if this is your reference on top with these 4 different regions, you may have this duplication or deletion, and that's kind of so the copy number variant calling will give you it will give you information about whether this event occurred, but not necessarily where it occurred. You can get more details using breakpoint tools, and you can actually say where those events exactly at which positions they occurred. Then you have inversions where part of your genome got flipped. Dispersions where it got copied or put somewhere else. Translocations where part of your genome from another contig came and get copied here, and very complex combination of these different things. And one of the problems with doing SV calling is how to standardize the description of all these events. It turns out to be not a trivial problem. And so right now there's a big pipeline for doing SV calling that came from collaboration between the Talkowski pipeline and MGH, and also our SV group from the GATK development team. And there recently came out a paper on this structural calling on the nomad, which I recommend you check out, and it's kind of like state-of-the-art structural variant calling right now at the moment. Yeah. So yeah, these are the different kind of pipelines that we will discuss in more or less details in the next few days. Finally, all of these pipelines you can find the workflows for them on this GATK GitHub repo called GATK workflows. It will have specific repositories for every particular type of pipeline. And also you can find them in the Terra workspaces, which we will talk about more today. Awesome. That's it. Are there any questions?